Hi, everybody. Welcome to Neuroscientists Talk Shop. Today is October 23rd, 2025, and we're talking to Dr. Peter Crino. Peter is the Richard and Kathryn Taylor Professor and Chair of the Department of Neurology at the University of Maryland School of Medicine.
Hi, Charlie. How you doing?
Hey, great to have you here, actually.
Thanks to be here.
So his clinical practice and research program includes studies of a broad landscape of brain developmental disorders, including drug-resistant epilepsy and autism, and there are some others, some of whose names I can't pronounce quite. And he's especially known for his studies showing the importance of a particular kinase, mTOR, and related pathways in a large group of these disorders. So also with us is Jenny Hsieh.
Hi, how are you?
Hi, Jenny. Jenny is our local expert on brain development and developmental brain disorders, and I'm hoping the two of them are going to take over the conversation pretty quick.
Sounds good.
I'm Charlie Wilson, your host. So there's a lot I'd like to hear you guys talk about, but I wonder if I can get you started by talking about these de novo somatic mutations and somatic genetic mosaicism during brain development. Because there are a lot of brain disorders that have an overt genetic inherited component. We recognize that. We think, we preferentially study those because we know the gene. But it could very well be that a lot of the ones that we say are not inherited are really just genetic disorders that have to do with somatic developmental disorders. So how widespread is that? What do we know about it? And is that as exciting as it seems from a clinical perspective?
Yeah, no, thanks for the question. So I wouldn't be so bold as to say it's a paradigm shift, but I will say that the identification of somatic gene mutations, which are de novo mutations that happen during embryonic brain development and result in a focal area within the cerebral cortex that is abnormal from a structural perspective and from a structural perspective, and which is probably linked to disorders like epilepsy, really has been a shift in our understanding of what causes epilepsy, kind of broadly speaking. A number of years ago, we really hadn't had the idea that these were somatic gene mutations. We had people who had epilepsy, seizures coming from the right frontal lobe or the left temporal lobe. And we didn't really have a reason for it. The identification of somatic gene mutations in these areas really gives us a target and an understanding of pathways that might be affected. It's a mechanism that accounts for different types of pathology in the brain, so either very mild pathology, but also some areas that have very, very severe disruption of the cerebral cortex. So the idea that the cause of these are actually just somatic mutations affecting variable numbers of neurons in the brain really has shifted our ability to understand kind of what causes focal epilepsy in many individuals.
And we usually think of the brain tissue as kind of immune to mutation because we don't think the cells are dividing, but there's a time when they do.
That's correct. Yeah,
and that's the critical point.
Yeah, so I think you hit the nail on the head. The time when cells are dividing are somewhere between human gestational weeks 8 and 20. And so it's very possible that during that time, there is a mutation that happens essentially in any dividing cell. Now, it seems as though most of the lineage of these cells are neuronal. We don't have any evidence that they're astrocytes or oligodendrocytes just yet, but it could be the case. Right now, the going idea is that these are neuroglial progenitor cells. They're undergoing active cell division. And like all dividing cells, they are susceptible to an error in DNA copying. And so any error in DNA copying that's not caught or edited results in a gene mutation. And so the numbers of cells that are affected can be very low, maybe 1 or 2%, but can also be very high, 50%.
And you mentioned during your talk that you can start seeing these regions of overgrowth on the fetal brain MRI, and you kind of alluded to that this could be a window of time for treatment. But is that already being done as a standard practice? And how do you? Is it dangerous or tricky to treat pregnant women?
Yeah, no, it's absolutely true. With the advent of fetal MRI, we used to do fetal ultrasound, but now we have the ability to do MRI. We can see these malformations early on. So the idea would be in the future, if there was a way to identify the somatic gene mutations, in theory, we could use targeted precision medicine to come up with something that would treat the malformation, maybe even prevent the formation of the malformation. It's possible that we could come up with something that would prevent the wiring of an epileptic circuit in the area of the malformation. Now, is it dangerous? I think it's going to very much depend on what the mutation is and what the actionable, pharmacotherapy is. I will tell you now there is a large consortium to look at prenatal sirolimus treatment in a number of brain disorders. And sirolimus, as you know, is an mTOR inhibitor drug. There's actually a group of investigators who are looking at it, and it's a multi-D group of neurologists, pediatric neurologists, maternal fetal medicine, OBGYN, looking at how tractable that strategy is and how safe it would be both for the baby as well as for the mom. Remember, any medicine that you use in the baby is not going to just go to the area of abnormality, but it's going to affect the entire developing brain. So we do have to be a little bit cautious. But no, I'm very excited about the possibility for preventative therapy in the prenatal time period.
So I remember when I was pregnant, ultrasound was standard, every several months. But I don't think I had anyone asked if I wanted to do a fetal MRI.
Right. Standard of care right now is to do ultrasound to look for major malformations, spinal cord malformations, anencephaly, other things that you might see. The decision to do fetal MRI is often made by either maternal fetal medicine specialist if there is some history of a genetic abnormality, if there's anything that's suspected on the ultrasound. So, for example, in the case of hemimegalencephaly, where one hemisphere is enlarged, often that's seen on the ultrasound, so the MRI is done to get a more high resolution picture. So again, the question becomes, do we want to prevent the malformation from forming, or are we thinking about preventing the sequela, which in this case would be seizures? And I think those are two different kind of approaches. But yeah, lots of people are thinking about this. We've done in utero treatment in mouse models of these, and the pregnant dams seem to tolerate it just fine, and we don't see tremendous effects on the pups. We have seen a runting effect in some of the pups with, for example, the mTOR inhibitor drugs, where the pups are a little bit smaller than their age mates, but they regain that over time. So I do think there's a strategy to be able to approach prenatal therapeutics.
But it's localized. The reason it gets localized in the brain is the mutation happens in a developing cell, and then its clonal offspring carry that mutation, and the rest of them don't.
That's correct.
And then that may happen more than one time in the brain.
That's right.
And so you could get more than one locus of this sort of thing. And so it's this explains the regional form of the brain malformation.
Yeah, that's right. So the idea is that you have a somatic gene mutation that happens amazingly in a single neuroglial progenitor cell. And that cell, by its very nature, will give rise to 2 more cells, and then 4, and then 8, and then 16. So you do have this clonal expansion where the mutation is carried forward with fidelity. So all of the daughter progeny of that cell will have this mutation. Now the issue is, why doesn't it just keep going and going and going? At some point it does stop, and the reason is we think that the cells maybe exit the cell cycle and decide to commit to a neural lineage. But it can be fairly sizable, and you can have a lesion that is, you can see it on the MRI. And as I mentioned in my talk, the allelic frequency, the number of cells who carry the mutation, can be very low, 1 or 2%, but it can be as high as 50%. The low variant frequency tend to be very small lesions. The high ones tend to be bigger hemispheric lesions.
So you're mostly thinking about cortex, and in the cortex, dividing cells form little spatially defined clones, but not everywhere in the brain works like that. Some brain areas, the cells divide, take off, and migrate, and spread out all over the place. So this, of course, you're studying this mainly in the cortex. You're studying epilepsy and diseases like that, but there may be a broader spectrum of diseases that we think of as more generalized, that are really happening from the same mechanism.
Yeah, oh, no doubt. So I think a very compelling response to that observation would be the paper from Chris Calley's group at Harvard, where they showed that some individuals who have temporal lobe epilepsy, just classic garden variety temporal lobe epilepsy, have mutations in MAP kinase pathway gene somatic mutations. And so even temporal lobe epilepsy, which we don't think about as a malformation, may actually result from a somatic mutational mechanism. We've had no ability to assay somatic mutations, for example, in the thalamus. Probably that's very relevant. We just haven't had the example and the way to do it. So my guess is, yeah, there's probably this mechanism is much more widespread than we're thinking. We do have a little bit of an ascertainment bias here, which is that the only tissue that we can study is the tissue that we take out during surgery. So that is a little bit of a challenge. I will tell you there are some initiatives now to look at the post-mortem brain and really look at what the spectrum is. It turns out that somatic mutations are fairly common throughout the brain. They're just tolerable. They don't cause anything problematic. The mutations that we're seeing are relatively intolerable because they're actually pathogenic. They're disease-causing mutations.
So they tend to get focused on these pathways that you study that are absolutely essential for controlling cell growth and formation of the tissue.
That's right. The mTOR pathway is a critical pathway for polarity, growth, cell size, maturation, differentiation, motility. So for the one group of disorders that I described today called mTORopathies, we do think the mTOR pathway is critical, and it does provide an actionable therapeutic target. For the other groups that are not the mTORopathies, it's a little more complex, and the genes are very heterogeneous. The genetic background and landscape is highly heterogeneous, and I don't think there's going to be a clear, easy fix with a one compound. We're going to have to do some more precision therapeutics to think about this.
So I was thinking about, you mentioned precision therapeutics at an earlier stage in your visit here, and I was thinking about, what it would mean to make use of this, because for example, if you take a skin cell from one of these patients, you're not going to see the mutation.
That's correct.
And so it's a little different from a real genetic inherited disorder like the kind Jenny normally studies, because she can take that patient's tissue, any of it, and it's all got the mutation in it. So what is precision medicine going to mean?
But even so, you can still take iPS cells and use gene editing to introduce the mutation.
We can, and we've done that. We absolutely have done it.
But in that case, there's no point in taking it from the patient, a particular patient.
Yeah, I think.
What do you think about the second hit?
So that's super complicated.
Hypothesis.
So the second hit hypothesis for tuberous sclerosis and NPR3 is very complicated. The condition of being heterozygous may just not be enough to generate a malformation. And so we really do believe in TSE for sure. We believe there's a second hit. We haven't found it in NPR3, but it's the same thing. There's another, by the way, there's another protein in that complex, DEPDC5, and then there's another one called NPRL2. They form a trimeric complex. And second hits actually have been found for DEPDC5. So we do think the mechanism probably is consistent. But you're right. If we were to do it in iPSC-derived cells, whether 2D or going on to organoids, you'd have to probably do a full knockout. You'd have to do a full knockout to actually achieve what you're trying to do. With CRISPR, the challenge, of course, is you have off-target effects and other possible things to control. When you do it from the patient, it's the actual patient variant that's in the stem cell. So that's a little more straightforward. We don't have a way yet to take cells from a somatic variant and turn them into iPSCs, right? We don't have that strategy yet. I'm hoping that will come at some point in time. We just don't have that mechanism right now.
Could you say something about what the two-hit idea is and how it's relevant?
Sure. So it's a great question. So the issue is for dominant disorders like NPR3 and tuberous sclerosis complex. In a dominant disorder, every cell in the body has inherited one mutated allele. So the question then is, if every cell has a mutation and every cell therefore is heterozygous, how do you get a focal abnormality in the brain? Like, what happened in that one region? The presumption, at least theoretically, and there is some experimental backup for this, is that there is a somatic second hit mutation in the other allele. And so you render all of those cells essentially null or knockouts. So what you have is an island of cells in the brain that are essentially null or knocked out, whereas the rest of the brain, the ocean, is essentially heterozygous. The other part of it is that the heterozygous condition doesn't confer a phenotype. And so the het is not enough to give you a phenotype. You have to have the full knockout. That's what the second hit gives you.
So if you're down one allele to begin with, it just seems like cruel fate that you would have a somatic mutation in that same gene, other allele. Do you think that's just a random event, or is it somehow being down one makes you more likely.
So, Knudsen posed this years ago in retinoblastoma. He asked the question, how can a kid who's got a genetic mutation in the RB gene have a focal tumor in one eye? How's that possible? And the idea he proposed was this second hit. What's interesting about second hits, and it's well reported in TSC, NF1, NF2, von Hippel-Lindau, any dominant disorder that has a focal manifestation, is that there's something about the allelic variant that engenders or induces or augments the likelihood of a mutation, not randomly in the genome, but in the other allele. And nobody really understands that mechanism. It's not clear. People have done genomic analysis and have not found mutations elsewhere. It's always in just the other copy of the gene. So you get a functional null state, essentially.
When you get a flat tire, it seems like that's when you're spare. It's flat.
Yeah.
That's it.
It's kind of a good analogy. It's actually a great analogy. Yeah, it's exactly right. So that mechanism has been well shown in TSC. And usually what you have is the germline is a larger deletion, and the second hit is a small single base pair error. It's usually not the other way around. But you can have the germline be a small base pair and the other one be a small base pair too. Anything that compromises the function of the gene-encoded protein.
So in your Mennonite, and I always pronounce Mennonite population study, I remember the patients had the same mutation, but the phenotype was very highly variable. And so I think, how do you think you can explain that? And also the ages were span like 1 to 37 years old.
Procedures, yeah. So that is a great example of the three things we think about with genetic diseases, which are one is penetrance, the other is expressivity, and the third is something called pleiotropy. And these are three common phenomena seen in genetic disorders. Expressivity is basically any one sort of, when you look at the phenotype of the patient, like how severe is that? So is this take epilepsy, for example. Is it that the child had just 2 seizures, or is this drug-resistant epilepsy? That would be different expressivity, right? Penetrance is how many people who have the variation actually express the disease. Some people there's only modest penetrance where maybe one of them has a seizure, one of them has ASD, but 2 other family members have basically nothing at all. The third is pleiotropy, which is that for any single gene mutation, you can have widely divergent phenotypes, even in the same pedigree. Even in monozygotic twins, you can have pleiotropy. So 2 twins who share the same genome might have different phenotypes, even though their genome is identical. That phenomenon is pleiotropy. And the truth is, we don't really understand what contributes. Is it the exposome? Is it random chance? Is it nutrition? It's just nobody really knows. TSC, tuberous sclerosis complex, this is well documented because there's large pedigrees. And in the same family, because TSC is a multi-system disease affecting brain, skin, eye, lung, kidney, heart, you can have this person who's got kidney, this person's got lung and kidney, this person's got epilepsy but no lung and kidney, this person's got lung, kidney, and epilepsy. So that's pleiotropy. And it's just a common phenomenon in genetic diseases. NBRL3 is interesting because it doesn't really seem to have anything other than a neurophenotype. There's no heart, there's no lungs, there's nothing else. So it's really just a neurophenotype, which is interesting.
So this is a mystery. Do we have any prospects for how these things can come about?
There's a lot of people who are working on how the other allele is stabilized, and that having one mutated gene depends on the mutation, right? If it's a big deletion, that leads to a fair amount of stability in the 2 alleles, right? So that's one possibility. For a missense mutation, I think it's up in the air. Nobody really knows how that seems to work. We do know this, that the other variant, though, doesn't seem to be a big deletion. So for whatever reason, it doesn't seem to be that. The 2-hit hypothesis in TSC has been shown in the kidney, in the lung, in the heart. So it's well described in TSC. There is a brain tumor that tuberous sclerosis patients get that this has been shown very clearly. The cortical lesions have been a little more challenging because when you take an entire cortical lesion, remember, as a somatic mosaic, you're going to get some cells with the second hit and some cells that don't have the second hit. So it's much harder technically just to detect the second hit. We did some work in my lab to do this, and we ended up sequencing both TSC1 and TSC2 genes, and we did it in single cells. It took me like 6 years to do this project. Other people can do something called LOH analysis, which is loss of heterozygosity, which is kind of like FISH, fluorescence in situ hybridization. You basically just look and see if the other allele is there or gone. It just, in the brain, it doesn't seem to be that you lose the other allele. It seems as though you just get a somatic second hit. Now, in NPRL3, We have not shown this mechanism yet. We've looked, we haven't found it yet. So it's still up in the air as to whether or not this is a second hit mechanism. I think it probably is, but we have not shown this yet.
Are there any structural or malformation for MPRL3?
FCD2. Yeah, so it's a focal cortical dysplasia gene for sure. All three of those genes, DEPDC5, FCD2, DEPDC5, MPRL3, and 2, all confer a focal cortical dysplasia type 2 pathology.
So patients with that mutation could have been candidates for the prenatal
Definitely
precision medicine, but knowing that there's this, interaction of and differences in penetrance, expressivity, pleiotropy, that's part of then for the patients and the family or the families to decide if this is going to be. It's a big choice
because the penetrance of epilepsy in this disease is actually only about 37%, right? So even if you have a brain malformation, you may not have epilepsy, which is super interesting. So if you were to detect a brain malformation on ultrasound or on MRI, you're going to decide, are we going to treat you to prevent seizures? Well, if the majority of people don't have seizures, that's a difficult conversation. I think for neurocognitive, it's going to be very interesting, especially for ID and ASD. Could you do something to improve or augment cognitive outcome? I think that would be very, very exciting to do that. But yeah, this is kind of some of the challenges. Unless it's fully penetrant and fully expressive, you have to decide, why would you be giving fetal therapy? Now, in Pretzel syndrome, which is another TOR-related disorder that's due to a mutation in a gene called STRADA, S-T-R-A-D-A, there the phenotype is big-headedness and intellectual disability with bad epilepsy. The penetrance of that disorder is about 100%. So in that, we actually did give 1/3 trimester mom sirolimus, and actually the baby's head came down in size. And we never published it because we didn't have an IRB, we didn't have a protocol. We did it as an expanded use, and we never published it, but we actually did it. So the mom was being queued up for a cesarean section, for a C-section, ended up not having a C-section, having a normal delivery. So we believe it can work. There was a paper published in the New England Journal of Medicine a number of years ago where they looked at cardiac lesions in TSC, and they treated the baby with sirolimus, and the lesions came down. The baby was in basically high output cardiac failure. They were going to have to do fetal surgery to take this lesion out to save the fetus. And instead, they just gave the mom sirolimus, and the baby survived. So the precedent for certainly third trimester fetal therapy, I think, is very solid. I think as you start creeping back into earlier second trimester, then I think it starts to get a little more challenging.
So all those examples that you just gave worked because those diseases are all over activity of mTOR.
Correct. That's correct.
And so you can just inhibit
mTOR. That's right.
So it almost sounds like just a switch. Either mTOR is overactive or it's not. But mTOR activity is probably graded and can go through
it's definitely graded.
So what is are there mutations that make mTOR underactive? And
not usually. So most of the mutations that happen in the mTORopathies are activating mutations in mTOR itself in a specific domain. There's a kinase domain and an FAT domain. Both of those groups are activating. And then the others are usually activator mutations in like AKT or inhibitor mutations in negative controllers. So DEPDC5, MPRL3, and MPRL2 are all inhibitors of mTOR. So loss of function variants confer hyperactivity to mTOR. But yes, there are some activating mutations. It doesn't matter. The net effect is that the kinase is hyperactive. The other final part of it that's a little complicated is that mTOR obviously has two isoforms, mTORC1 and mTORC2. Much of my remarks today were confined to mTORC1, which we think is the primary, but mTORC2 is not without significance, and the drugs sirolimus and everolimus don't really have much efficacy at mTORC2. You need a different pharmacotherapy. So that's maybe part of the reason why the drugs have not been as fully efficacious as we think they might have been.
I have to ask you about the possibility of somatic mutations being responsible for personality features and people's special capabilities or lack thereof. I can't play the piano for beans. Is that Is that because I didn't get the right mutation?
So again, not all mutations necessarily are deleterious, right? The very foundation of evolution is that mutations can confer advantage as well. So nobody has shown that yet. It's purely theoretical, but the group that is together to talk about somatic mosaicism, it certainly has crossed the table in discussion. Is, for some things, is it possible that there are mutations that confer augmented abilities? Or for people with dyslexia, for example, maybe it's not a germline thing, it's a somatic thing. Many people now with ASD, we're doing whole genome sequencing. We're not finding anything. So do we say that it's an exposome thing? Or is it possible that it's just somatic mutations in the brain? I think that's the next generation of investigation. Remember, in order to do this, though, there is the challenge. You have to have brain tissue to do this. And so you've got to be looking in the brain itself. We don't have a proxy for the brain yet.
Yeah. And you just can't take a piece of my brain
that
you think is my piano playing part of my brain.
Well, maybe we would take the piano playing part because it wouldn't affect you.
It wouldn't have any effect on me at all. Well, okay. Thank you very much, Peter Crino and Jenny Hsieh.
Thanks, Charlie.
And
pleasure chatting with you.
It's been really great.
Thank you.
This has been Neuroscientists Talk Shop.